Functional anatomy of the luring apparatus of the deep-sea ceratioid anglerfish, Cryptopsaras couesii, Lophiiformes Ceratiidae. The osteology and myology of the ileal apparatus of Cryptopsaras couesii were examined in an effort to elucidate function. The apparatus consists of two bones, a tiny ileal bone, completely enveloped by tissue of the esca, and an extremely long supporting pterygophore that lies within a deep groove on the dorsal surface of the head. The anterior end of the pterygophore emerges on the tip of the snout from between the frontal bones, while the posterior end, encased in a dermal sheath when the ileal apparatus is retracted, emerges on the back, just anterior to the dorsal caruncles. 5 pairs of muscle control movement of the ileal apparatus. Small, short erectors and depressors control movement of the ileal bone, while extremely long inclinators, protractors, and retractors control movement of the pterygophore. The protractors and retractors of C. couesii are more robust and much longer than those of other Lophiiformes, indicating that the pterygophore of this species has an exceptionally wide range of movement in the anterior and posterior plane. Moreover, these muscles wind around the pterygophore in opposite directions, a unique anatomy that suggests that C. couesii extends and retracts the pterygophore by rotation. Anglerfishes of the teleost order Lophiiformes are well known for their peculiar mode of feeding, in which the first dorsal fin spine is located out on the tip of the snout and modified to serve as a luring apparatus called the ilium. The morphology of the ilium and its terminal bait, the esca, varies considerably among anglerfish taxa, 18 families in 66 genera and about 300 species. The anatomical details of the esca, being species-specific, within nearly all members of the deep-sea suborder Ceratiidae. Cryptopsaras couesii gill, family Ceratiidae, is one of the commonly collected ceratioids occurring around the world oceans at depths ranging from about 75 to 4,000 meters. The majority of known specimens have been taken between 500 and 1,250 meters. Adult females of C. couesii are characterized by having an elongate pterygophore lying within a deep groove on the dorsal surface of the head and body and bearing distally a greatly reduced ileal bone. Bertelsen, 1951, described the osteology and myology of the ileal apparatus of a larval female C. couesii, but the anatomical details of this unique device in metamorphosed and adult specimens have never been reported. Here we describe the ileal anatomy of adults of C. couesii and discuss its function relative to other known Lophiiform fishes. Materials and methods. The ilium and associated muscles were stained with alizarin red S and dissected and illustrated with the aid of a dissecting microscope. Terminology describing external anatomy follows that of Bertelsen, 1951, Peach, 1986, and Bertelsen and Kreft, 1988. Osteological and myological terminology follows that used by Bradbury, 1967, Winterbottom, 1974, and Peach, 1981. Standard length, SL, is used throughout. Symbolic codes for institutions are those provided by Leviton et al., 1985. Species examined: Cryptopsaras couesii, 7 of them; Loftius liliton, 1; Antenarian striatus, 1; Brachioniphysis, 1; Chonax abii, 1; Dibranchus japonicus, 1; Melanocytus johnsoni, 1; Buferaceus thelli, 1; Onerodes thomsoni, 2; and Linophrine densiramus, 1. So they are identified by code and by length in millimeters of standard length. Results: external anatomy: pterygophore of ilium long and slender, lying horizontally within a deep groove on dorsal surface of head, emerging anteriorly from interorbital region, figure 1a, length of anterior extension 39.1 and 88.3% standard length in two of the specimens respectively. In 126 millimeter specimen, posterior end of pterygophore emerging on back just anterior to largest of three dorsal caruncles, see Bertelsen 1951, encased in a sheath of skin, figure 1b. In remaining specimens with pterygophore fixed in a more anterior position, dermal sheath of pterygophore withdrawn into deep groove, bounded on each side by epaxial musculature, figure 1c. Iliceal bone minute completely enveloped by tissue of esca, figure 1a. Esca small, oval in shape, figure 2. Proximal 1/2 to 2/3 of the escal bulb darkly pigmented, distal part covered with translucent skin, apex darkly pigmented, a single distal escal appendage, trifurcated at base, central branch further dividing into many branches. Figure 1, Cryptopsaras couesii, one specimen. A close-up photograph of dermal sheath in this specimen. B, and this specimen. And C shows a caruncle and an esca. EDS emerging dermal sheath, and WS withdrawn dermal sheath. Figure 2, left lateral views of escae of Cryptopsaras couesii, one specimen and another specimen. B, bars 1 millimeter. So they almost look like a bulb with little stalks coming out of it. Figure 3, lateral views of Elysium, a Cryptopsaras couesii, Lissa specimen, 100.9 millimeter in pteryogophore length. B, Antenaria striatus, Illis specimen number. Onerodes thomsoni, a specific specimen. 1b is the iliceal bone. PTR is the pteryogophore of Elysium. SII is the second dorsal spine. The bars are 5 millimeters. Figure 4, iliceal bone of Cryptopsaras couesii, a specific specimen, in lateral A, frontal, B, and rear C views. The bars are 0.5 millimeters. Osteology and myology. The following osteological description is based on the 106 millimeter specimen, and the myology is based on the 39 and 93 millimeter specimens. Iliceal bone, figure 3a, extremely small, 0.8 millimeters in width, 0.9 millimeters in height, completely enveloped by tissue of esca, proximal part short and thick, attached to anterior tip of pterygiophore by connective tissue, distal part separated into right and left halves by anterior and posterior notches, and deeply concave on dorsal margin, figure 4, pteryogophore of Elysium, figure 3a, extremely long, 101 millimeters, 95% of standard length, uniformly thin. Anterior end emerging from between frontal bones on dorsal surface of head, extending anteriorly well beyond mouth, central part lying within deep groove bounded on each side by epaxial musculature, figure 5. Posterior part connected to medial surface of dermal sheath by soft connective tissue, protruding from back just anterior to dorsal caruncles, figure 1b. Movement of pteryogophore controlled by 5 pairs of muscles, erectors, depressors, inclinators, protractors, and retractors, as described in part by Brouwer 1908, a pair of erector muscles originating from lateral surface of posterior part of pterygiophore, figure 6a, running anteriorly along pterygophore, figure 6b, inserting on anterolateral surface of iliceal bone via an extremely long and thin tendon, figure 6c, a pair of depressors originating from lateral surface of posterior part of pterygophore just above origin of erectors, figure 6a, running anteriorly along length of pterygophore and abreast with erectors, inserting on posterior surface of ilioceal bone via an extremely long and thin tendon, figure 6b and c, a pair of long, thin inclinators, figure 5, originating from lateral surface of lateral ethmoid of cranium, inserting on fascia of retractors, figure 6a, a pair of protractors, figures 5 and 6a, originating from anterodorsal surface of frontal bone, inserting on lateral surface of posterior tip of pterygophore, where the muscle is wound around the shaft of the pterygophore, twice in this specimen and 4 times in another specimen, figure 6a, a pair of retractors, figures 5 and 6a, originating from lateral surface of post-temporal bone, inserting broadly on central part of pterygophore, the latter muscle extending medially on dorsal margin of epaxial musculature into ilioceal groove on top of head, figure 5, changing course abruptly anteriorly, emerging from anterior opening of groove, and winding around pterygophore in opposite direction to that of protractors, twice in one specimen and another specimen, figure 6a. Figure 5. Dorsal view of head of Cryptopsaras couesii, a specific individual. Pterygophore of Elysium, INCL is inclinator and PROT is protractor, RET is retractor. The bar is 5 millimeters. Figure 6. Lateral view of Elysium muscles, posterior A, middle B, and anterior part C in Cryptopsaras couesii, a specific specimen. Antenarius striatus, and on Neurodes thomsoni, each a specific specimen. ERE stands for erector, DEP for depressor, IB for ilioceal bone, INCL for inclinator, PROT for protractor, RET for retractor, TDEP is for tendon of depressor, and TERE is for tendon of erector. The bars are 1 millimeter. Oh wow, so in the first picture in figure A, you can basically see like all of this stuff is happening. The protractor, retractor, etc. And the Elysium. That's wild. There's a lot going on. So the Elysium has a bunch of muscles that allow it to move in all of these different directions. Discussion. In most Lophiiform fishes, the ilioceal apparatus, usually situated on the dorsal surface of the skull, consists of a supporting pterygophore and 2 dorsal fin spines. The first spine, called the ilioceal bone, is usually well developed and bears the terminal bait or esca. The second spine is nearly always highly reduced, lying on the dorsal surface of the pterygophore, just behind the base of the ilioceal bone, figure 3b and c. Movements of these bones are controlled by 5 pairs of muscles, 3 of which are responsible for movements of the pterygophore. The retractors and inclinators run between the second dorsal fin spine and the skull, while the protractors run between the pterygophore and the skull. Anterior and posterior movements are controlled by the retractors and protractors, and left and right lateral movements are controlled by the inclinators. These movements appear to be limited because these 3 muscle pairs are relatively short in length. The remaining 2 pairs of muscles, the erectors and depressors, are rather well developed and connect the ilioceal bone to the pterygophore, figure 6DE, thus controlling anterior and posterior movements of the ilioceal bone. In contrast, the ilioceal pterygophore of Cryptopsaras couesii is unusually long and lies within a deep longitudinal groove on the dorsal side of the skull, formed by right and left epaxial musculature, figure 7. The ilioceal bone is extremely small, while the second dorsal fin spine is absent, see figure 3a. Judging from this anatomy, and as previously described by several authors, e.g. Bertelsen 1943, The ileal apparatus of C. couesii can be extended far forward, well beyond the anterior margin of the jaws, and likewise retracted backward to take on an extreme posterior position relative to the cranium, figure 8. These movements are controlled by the same 5 pairs of muscles previously described, but in C. couesii, the erectors and depressors are extremely weak, inserting on the ileal bone via long tendons, figure 6c. Therefore, it appears that movement of the ileal bone of C. couesii, figure 9bd, is rather limited when compared with that of other Lophiiform fishes, figure 9a and c. Of the 3 muscle pairs related to the movement of the pterygophore of C. couesii, the inclinators are unusually thin, undeveloped, but extremely long, see figure 5. In contrast, the inclinators of other Lophiiformes examined are quite short and therefore responsible for only slight anterior and posterior movements of the pterygophore, figure 9c, whereas the pterygophore of C. couesii has an exceptionally wide range of anterior and posterior movement, figure 9d. The protractors and retractors of C. couesii are more robust and much longer than other muscles, figure 6a. The retractors are especially long and complex, figure 5. In addition, the protractors and retractors run along the length of pterygophore, spirally winding around it, figure 6a. A similar condition of the protractor was found in the larger specimens of Ceratis hobeli examined here. Bertelsen, 1951, assumed that this condition gave a better hold on the very thin pterygophore. Therefore, conditions of the protractor and the retractor, recognized here, also suggest a similar ability. Although most of the work described here supports the findings of Bertelsen, 1951, and others, no one heretofore has mentioned the peculiar winding of the elongate retractors and protractors around the pterygophore in opposite directions, figures 6a and 9b. This unique anatomy possibly suggests that Cryptopsaras couesii extends and retracts the pterygophore within the ileal groove by rotation, figure 9d. To our knowledge, this rotational biomechanical mechanism is unique among fishes, if not among vertebrates in general. Figure 7. Schematic diagram showing relation of pterygophore and its related muscles in Cryptopsaras couesii. A. Dorsal view. B. Lateral view. Abbreviations as in figure 5. Figure 8. Schematic diagram showing movements of ileum in Cryptopsaras couesii. A. Protruded position. B. Retracted position. Abbreviations as in figures 1 and 3. Figure 9. Diagrammatic lateral A and dorsal C, D views of ileum showing bony elements and muscles of ileum in general Lophiiform fishes A, C, and Cryptopsaras couesii B, D. Black arrows indicate direction and contraction of muscles. Dotted arrows indicate rotate movement of pterygophore. Patterned arrows indicate movement of ileal bone and pterygophore by erector, M-ERE, depressor, M-DEP, inclinator, M-INCL, protractor, M-PROT, and retractor, M-RET. Osteological and myological abbreviations as in figures 3 and 6. So A is showing all the stuff close up. You can see all the muscles, the skull, and the muscles around everything. And figure B, oh, that's so interesting. You can see how it's attached to the skull and the apexial muscle behind it. And figure A is showing how the ileum basically moves out and then retracts back in. Figure 9 is basically trying to show that things are rotating and just how all the protractor, retractor, etc. are moving.